Recently, the International Society for Stem Cell Research, ISSCR, released Standards for Human Stem Cell Use in Research, a document that outlines a set of recommendations that establish the minimum characterization and reporting criteria for working with human stem cells. The overarching goal of these criteria is to standardize practices and in doing so improve the quality of results. But there are still no agreed upon minimum acceptance criteria for the functional activity of iPSC-cardiomyocyte models. For over a decade, human-induced pluripotent stem cell-derived cardiomyocytes have been widely used for safety pharmacology and disease modeling studies. However, it is left to the end user to determine whether the electrophysiological phenotype of their cell model is fit for purpose. The multielectrode array MEA field potential assay has become a popular tool for characterizing the electrical activity of iPSC-derived cardiomyocyte batches, studying cardiac disease models, screening for new therapeutics, and evaluating drug-induced cardiotoxicity. The goal of this project is to set the minimum acceptance criteria for a spontaneous beating wild-type iPSC ventricular cardiomyocyte field potential assay for compound testing and/or disease modeling. Using our own experience with iPSC cardiomyocytes in our respective labs in academia and in industry, our work with international consortia developing iPSC cardiomyocyte assays, SIPA and JICSA, combined with published data, we've developed the Axion iPSC Model Standards, or AIMS framework. This is a proposed standard focused on the iPSC cardiomyocyte spontaneous beat rate, features of the cardiac waveform, depolarization spike amplitude and field potential duration, and the synchronization of activity in the syncytia. It is important to stress that the standard proposed here is not intended to define the ideal cell activity, but rather to establish the minimum acceptance criteria for the functional activity of iPSC ventricular cardiomyocyte models. In addition to cell functional activity, other important criteria related to cell quality, for example, cell viability, sterility, absence of mycoplasma, can have a significant impact on cell activity. However, the methods used to measure these characteristics and determine associated acceptance criteria have been reviewed elsewhere and fall outside of the scope of the AIMS framework. Defining the minimum acceptance criteria for a spontaneous beating, wild-type, iPSC ventricular cardiomyocyte field potential assay for compound testing and/or disease modeling. The review team agreed that for an iPSC cardiomyocyte cell source to be considered as meeting the standard, at least 80% of the wells seeded with iPSC cardiomyocytes would need to meet the acceptance criteria. 80% was set as the threshold because lower than this level makes the cost of running the assay prohibitively expensive. In other words, if using a 96 well plate, 77 or more wells would need to meet the acceptance criteria. Cell models where less than 80% of the wells meet the acceptance criteria could be useful for preliminary exploratory studies, but would be impractical for use in an assay. So now for the acceptance criteria. For a well to meet the proposed functional standard, the iPSC cardiomyocytes need to meet all four of the following well acceptance criteria. 1. A spike amplitude of greater than or equal to 0.5 millivolts on 50% or more electrodes in a well. 0.5 millivolts was set as the threshold for spike amplitude, as this allows for reliable capture of spontaneous iPSC cardiomyocyte activity in baseline and could also accommodate a significant compound-induced decrease in spike amplitude. For studies focused on compound-induced effects on spike amplitude, however, a spike amplitude of greater than 1 millivolt would be preferable to provide a larger assay window. 2. Synchronized beating across 50% or more electrodes in a well. Electrodes in the array are considered to have synchronized beating if within a window of 30 consecutive beats, consistently, the first and last beat across electrodes occur within 30 milliseconds of each other. The synchronized beating should initiate from a single point of origin, where the inter-electrode beat time delay increases monotonically as a function of distance from the origin. More than one patch of spontaneous beating iPSC cardiomyocytes in a well is undesirable and is indicative of a poorly coupled syncytium. The competing spontaneous activity can confound the analysis of the results, Consequently, if a well has 2 or more independent patches of spontaneous beating iPSC-cardiomyocytes in the baseline recording, this well should be discarded. 3. A spontaneous beat period of 750 to 3000 milliseconds, or in other words, 20 to 80 beats per minute. The acceptable beat period range is wide. However, the rationale for this range was inferred from the SIPA pilot study, which used iPSC cardiomyocytes from 2 vendors with spontaneous beat periods at the 2 extremes of this range. See Miller et al. 2018, Figure 2. Although the spontaneous activity of these 2 cell types differed significantly, consistent concentration-dependent effects were observed for both cell types. See Figure 5. For the top end of this range, working with iPSC cardiomyocytes with spontaneous beat periods greater than 3000 milliseconds can be impractical due to the longer recording periods required to capture sufficient beating events for analysis of these datasets. It is worth noting here that primary ventricular cardiomyocytes should not spontaneously beat, and the spontaneous beating observed in iPSC ventricular cardiomyocytes is often attributed to their immaturity. Efforts to enhance the maturation of iPSC ventricular cardiomyocytes are ongoing and can result in spontaneous beat periods greater than 3000 milliseconds. For assays involving these iPSC cardiomyocytes, external pacing via electrical or light-induced means will probably be required to make these cells more practical to work with. A paced iPSC cardiomyocyte assay could be the subject of a future AIMS, but it is beyond the scope of this standard. Note, this standard, AIMS CM01, is specifically for iPSC ventricular cardiomyocytes and not iPSC atrial cardiomyocytes. Studies have shown that iPSC atrial cardiomyocytes can often have a spontaneous beat period less than 750 milliseconds. See Thorpe et al. 2023, Figure 2. 4. A corrected field potential duration of 200 to 700 milliseconds, where the field potential duration is corrected using the Federici correction. The acceptable corrected field potential duration range is wide. However, the rationale for this range was inferred from the SIPA pilot study, which used iPSC cardiomyocytes from 2 vendors with corrected field potential durations at the 2 extremes of this range. See Miller et al. 2018, Figure 2. Although the corrected field potential duration of these 2 cell types differed significantly, consistent concentration-dependent effects were observed for both cell types. See Figure 5. An additional point to consider: spontaneous beat regularity. Regularity of spontaneous iPSC cardiomyocyte beating was identified by the review team as an important factor in the quality of iPSC cardiomyocyte recording. However, this parameter, sometimes measured as coefficient of variance of the beat period, is less commonly reported in the literature. See Clements and Thomas, 2014, Figure 3, for an example of how this can be measured. Consequently, the review team did not feel comfortable setting an acceptance criteria relating to beat regularity at this time, but this will be something to consider should this AIMS be refined in the future. Many cell vendors use the spontaneous electrical activity of iPSC cardiomyocyte batches as measured with Maestro MEA as release criteria for their cell products. Here, cell performance data from 3 leading commercial sources of iPSC cardiomyocytes are presented with respect to this proposed standard. iCell cardiomyocyte squared from Fujifilm Cellular Dynamics Inc., Axo human iPSC-derived ventricular cardiomyocytes from Axol Bioscience, and Cardiocyte S from Nexcel. As you can see, the AIMS CM01 standard accommodates all 3 cell sources. Finally, it is important to consider the cell culture protocol. The reproducibility and quality of the iPSC cardiomyocyte model's functional activity is dependent on the cell culture conditions. Consequently, participating cell vendors must specify key details in their recommended protocol. These include temperature of the environmental chamber in the Maestro MEA system, CO2 concentration in the environmental chamber, cell culture medium that the MEA recordings should be performed in, when the cell culture medium should be refreshed prior to the MEA recording, the equilibration time given between docking the MEA plate and the start of the recording, the experimental window, that is, the specific time frame within which the iPSC cardiomyocytes will perform as claimed, and recommended appropriate positive and negative controls for the response of interest, with expected responses detailed for the former. For example, application of the hERG potassium channel blocker E4031 at 100 nanomolar should result in a greater than or equal to 20% increase in the corrected field potential duration. In summary, here we've established a functional standard for iPSC-cardiomyocyte electrophysiology using the MEA assay, our goal being that this new standard will help advance the adoption of iPSC-cardiomyocyte assays by aiding data comparison and validation. To learn more about this AIMS project, scan the QR code to visit our website for more information about AIMS, iPSC model standardization, and provide feedback.